believe it. I'm so really moved to be in this room with you today. I really want to thank you. I'm so grateful that you're here, and I'm really excited about the day, but we have a lot to cover in a short time, so I want to move on. I'm just going to ask a couple things, is that if, for families, we have children, people with whatever you need, if you need to leave the room for a drink or use the bathroom, whatever, you can go out the hallway and come in. The other thing I just ask, if you're going to speak, if you could use the microphone, please, because you know how hard it might have been for you to get here today. There are other people who actually couldn't make it here today, and we want to make sure to have them feel included. So we're going to use the microphone. I was thinking about doing introductions, but we kind of need to be quick because we have a lot to cover in a short amount of time. So I'll just pass the microphone if you want to just introduce yourself and say something about you or your family.
Hi, I'm Adam. I'm Sarah's son. She's pretty awesome. That's probably how most of you would know me. That's about it.
My name is Lorene Urquiza. I am a patient with titinopathies and had been misdiagnosed with inclusion body myositis for 20 years. So I just figured this out.
Hi, I'm Pat Foy. I'm Sarah's husband, Adam's dad. I'm a physician in New Jersey, but not in this particular area. Rachel did ask me to just announce, that and Sarah probably said this already. This is a session related to Titan. So if you're more interested in something other than Titan, then there are other spots for you too. But we're just really psyched to have everybody here.
Hi, I'm Stephanie Olson, known as Sarah's sister. I carry the same mutation in Titan as Sarah does, and both my children also carry the mutation. And I'm more of a carrier and very mildly symptomatic.
Hi, I'm Matthew Slisher. I have a daughter with Titan.
I'm Alyssa Slisher. Our daughter is 6.5. Her name is Lyra. Hi. We are both carriers of 2 different mutations. I am a partially affected carrier of a neutron mutation. Welcome.
My name is Mary Fitzpatrick. I'm new to the whole Titan way of thinking, I guess, the science of it all. I was diagnosed in February with cardiomyopathy and left ventricular non-compaction. Came to find out from a cardiac geneticist that both my brother and I, who both have cardiomyopathies, have the same variable of unknown significance on the Titan gene, which our genetic cardiologist said is the Titan, and that's causing our heart issues. So I'm here to learn, actually, from all of you.
Hi, I'm John Smith. I do research on Titan in Tucson, Arizona, and I've been really impressed with this meeting so far. I'm looking forward to today's session.
Hi, my name is Heather Gottlieb. I'm from San Diego, where my son Mitchell, he's 13, has a Titan gene myopathy. So he's in a wheelchair, can't walk. So I'm just here to learn and meet some people that are like my family.
Hi, I'm Sarah Newhouse. I'm a clinical fellow at NIH. I work with a lot of the people here, and I'll be talking more tomorrow.
I'm Kim Koto, my husband Adam. We both are carriers of a variant, and our son was lucky enough to get both of them. So we're kind of new to this as well and learning about as much as we can. You want to say hi?
I'm John Lovett. I'm here with my grandson Asher, my daughter Maggie, and my wife Louise. Asher's been diagnosed with variances on the Titan gene.
I'm the daughter of him.
Hi, my name is Taylor Nygaard. I'm a patient. My father has dilated cardiomyopathy. My brother passed of sudden cardiac death, and I'm symptomatic of cardiac and skeletal issues.
Hi, I'm Dale Nygaard, also known as Taylor's mom, and I'm just very grateful for you guys putting this thing together to help us. Thank you.
We're Zach and Taylor Werner, and we're here because our son was diagnosed with what is it? Congenital Titan myopathy, and we both are carriers of Titan gene.
Hi, I'm Megan Leach, I'm a nurse practitioner in Oregon, and the end. Welcome everybody to this session. The idea is to make it very flexible and to make it very useful for you. So we are here to answer to your questions in as much as we can, but first all the speakers are going to introduce themselves, and then maybe we're going to present a brief overview of what is proposed at Emory University as resources and for diagnosis, and then we can give an overview of the diagnostic process, and then we're here to answer your questions. Oh, yes, of course, sorry. I'm Ana Ferreiro. I'm a neurologist specialized in congenital muscle conditions in Paris at the Institute of Myology, and I'm also the head of the research lab that works on understanding the causes of congenital myopathies, and we were lucky enough to identify the first forms of congenital titinopathy in the first families. So I know very well how difficult it is to be without a diagnosis, how difficult it is not to know what is causing your disease or your child's disease, and how difficult it is to be a pioneer in novel forms of congenital disease that we are just learning to understand.
My name is Sandra Donkervoort, and I'm a genetic counselor who works at the National Institute of Health with Dr. Karsten Bonneman. We're involved in seeing many of the patients that have the muscle phenotype related to recessive titinopathies, and we collaborate a lot with Emily Oates in establishing the spectrum, the phenotypic spectrum related with recessive titinopathies.
And I'm Dimah Saade. I'm a pediatric neuromuscular neurologist, and I'm a clinical research fellow on Dr. Bonneman's team. I work with Sandra and Sarah Newhouse, and we are involved in the imaging, diagnostic, phenotyping of patients with a wide area of neuromuscular disease, but also specifically relevant to this session, titinopathy.
Hello everyone, my name is Samya Chakravorty, and I'm a clinical genetics researcher in Emory University. First of all, I must say that I'm privileged to be here. This is the first time I'm in a scientific and family conference where all these researchers that we work for is for you guys, so I'm being able to see you guys, so I'm really privileged for that. And so I will be talking a little bit about my new endeavors in Emory about enhancing molecular diagnostics, because from our experience, at most, or at least 50% of the patients remain undiagnosed, in not only titinopathy, all across the board in neuromuscular. So it's important to get the first thing done and enhance the diagnostics, so that's what I'm going to talk about. Thank you.
And I'm Emily Oates. I'm from Sydney, you can probably tell from my Australian accent. I'm a clinical geneticist, and in Australia, clinical geneticists also manage patients quite actively. So I've worked in the neuromuscular field for the last 10 years, and I have a particular interest in titinopathies, particularly the congenital form of the disease. And I'm absolutely stoked to be here today.
Well, thank you all. So maybe you want to start presenting the resources, and then we take it from there with Emily to give us an overview of what we know about the diagnosis pathway, or the pathway to diagnosis, rather, that can be really lumpy. And so please, Sandy.
Thank you. Hello everyone. Just to let you know that why I did say this new genomics to improve diagnosis of neuromuscular disease. So in the research field, we use a lot of functional tools, functional experiments and assays, which we don't really use in the clinic, diagnostic clinic. So our goal is to enhance the diagnostics by using these tools and push it through to the clinic, so that all this diagnostic odyssey and this 20 years of misdiagnosis can be stopped, especially with newborn screening coming up with all these children. So it's important to have all these tools in the clinic so that we can enhance the diagnostics. And that's the first thing that you do in a neuromuscular in the neuromuscular disease field. So just to give you a little bit of overview is we have been doing a great job in discovering genes, rare disorders related to rare genes in the neuromuscular field, and you can see the increment of gene discovery in the muscular dystrophy field. But that causes a problem also. where we don't know the variants that we find in those newly discovered genes are disease-causing or benign, right? So that those patients remain undiagnosed for years. So at least 40 to 50% of all across the board, all muscular dystrophy, including titinopathy, remain undiagnosed. And this is, and when we are talking about clinical genetics field, we don't call mutations, we call variants, and the variants can be classified either disease-causing, which is pathogenic, they're called as pathogenic, or it is benign. Or sometimes if we don't know if it is pathogenic or benign, it is called variant of uncertain significance, or VUS, which is a big diagnostic burden in the field. If you have a VUS, you cannot get recruited to clinical trials. You have to classify those VUS either pathogenic or benign to have the complete diagnosis. Otherwise, you cannot recruit patients to clinical trials or precision medicine regimen. And this is our Emory experience. This is a chart from all disorders, cancer, neuromuscular, everything. Okay, and if you can see the burden of this variant of uncertain significance, right? So, and then when you look at our cohort, we actually, for the last 2-3 years, we sequenced almost 5,000 patients, and we have been able to diagnose 30% of them by 35 gene panel sequencing. So it's called next-gen sequencing, DNA testing. But the problem is, when you look at in our cohort, it's similar. Like the burden of the variant of uncertain significance is similar to all disease cohorts. So there is a problem that we need to actually target and enhance diagnostics. So as I said, like you can, if you have undiagnosed patients, or if the VUSs are not classified, you cannot recruit them to precision medicine or other clinical trials. Now, Titin, in our experiments of that big 5,000 patient panel sequencing effort, we found that in Titin there are lots of number of VUSs. Actually, Titin is one of the least contributing among all the LGM limb-girdle muscular dystrophy patient cohort of all the 5,000 patients. But interestingly, they have the largest number of VUSs, which means all these VUSs needs to be classified. And also, we are actually, the titinopathy is underdiagnosed. It's underdiagnosed. So we need to classify these variants and enhance the diagnosis of titinopathy. And another problem that we face is sometimes we see single variant in Titin and another couple of variants in some other gene. Those patients also remain undiagnosed. So along with the VUS problem, the multigenic, so multiple genes are affected along with Titin. So that's another big diagnostic burden. And you can see that in our cohort, 30% of all those 5,000 patients have this type of variants. So those also needs to be classified using more functional tools which we use in research. We have to use that, keep on using that. By using patient samples, we can push those tools into the clinic for enhancing diagnostics. And as I was saying, like for example, these are some of these patients' clinical testing data that we found that there is a pathogenic variant in Titin. It's disease pathogenic meaning disease causing a variant in Titin, but also at the same time there are some other genes that are affected. Some are pathogenic, some are VUS, which needs to be resolved as well. So the way I'm doing it is having sort of a big omics, sort of a whole system-wide approach, where once you have DNA testing done, if it is not diagnosed, we can look at the RNA, the protein, the metabolites, and if sometimes create animal models to first of all classify those variants and give the research report back to the patients and the physicians for molecular diagnostics. But also we want to understand if there are biomarkers specific to the disorder. So in that way we do that. And just to let you know that there are, I can provide, if there are undiagnosed patients, I provide research reports in this format. This is a dysferrin case where we found this, pathogenic genetic events, 2 pathogenic genetic events dysferrin gene. Similar thing can be done in Titin as well. And this is another thing that I want to let you know, that there is free testing available. So this type of testing is also free. You can get in touch with me if you know some families who need diagnosis, or if your VUS is not classified, I can help you with functional assays free. And also there is all neuromuscular and all cardio gene panel sequencing free testing being available through PerkinElmer and Emory collaboration, which is called the Lantern Project. So if you just Google Lantern Project, there is free testing available. Spread the word out. Any limb-girdle muscular dystrophy or any cardiomyopathy panel, so all free testing is available there. So, but once that free testing is done, if you're still undiagnosed, you can still come to me in Emory, and I can provide a research report by doing the assays, and we'll try to diagnose the patients. So I think that's it. If you have a question, let me know, please. Yes. Yeah, Dr. You have a question?
Dr. Chakravorty, do you allow for parental classification, like to see if it segregates with each, if a single mutation segregates with each parent for free?
Yes, absolutely, and that's very important. That's a very important point. Whether it is we have to know whether, because Titin can be autosomal recessive or dominant, so we have to do that. Sometimes we are unlucky in the sense that some of these patients are later onset, so their parents have died already. So, but I mean, if it is available, we will definitely do that. Yeah.
Any other questions?
Yes, the example that you showed for the VUS for Titin, it also showed some other genes, which possibly might be something that modifies the severity of the condition. Would that also be made available to the patients?
Yes, absolutely. So the research report will have clinical correlation as well. So once whatever we find with functional assays and the genetic test report, we will be correlating with the clinical data, and that's why yesterday I talked about having the clinical data available to us. Then we can correlate that, and that will be in the research report. Yes. So the idea is to have a complete genotype-phenotype correlation as much as possible.
Thank you. So he said free like 10 times, right? The other thing is that you did answer the question that I had to Dr. Jonersman, which is, do you do testing of additional family members? But if the first person proband testing had been done either in a research lab or at a private clinical laboratory, but additional family members wanted to get testing but weren't sure what to do or didn't have the funding to get the testing, would you do additional familial segregation testing if you didn't test the proband? That which would be the initial case, for example.
Yes, so one of the perks I have is that I have a good industrial collaboration. So this Lantern project is headed by my previous postdoc boss. So in that way, there is a possibility that, I mean, of course, we cannot sequence the entire family of 6 or 7 people, but if there are parents available, we can definitely sequence it for free through the Lantern project, and I can analyze that.
Thank you.
So just a plug for when you do the genetic testing, we always advocate to make sure you do the whole, like, not only the genetics, but take the whole picture into account. So also, the genetics, of course, is a very important piece of information, but you want to correlate that with clinical presentation, family history. So not only look at genetic testing in isolation, but maybe pursue it with a genetic counselor, with a neurologist who can correlate it clinically, since there is so much that we don't know about yet.
Yeah, whatever testing we're doing, that's why I put that there, clinical correlation. So, I don't know if it's on. Oh, she's testing. So, this clinical correlation is, and from our experience, we have seen that we didn't have the clinical data, and we actually diagnosed for a different gene, which was misdiagnosed. So, if we had that clinical data, we could have focused on specific sets of genes. So that's the ability.
Can you hear me? I just wanted to make a quick comment. I think we talked a little bit about Titin interacting with other genes and that possibly modifying the phenotype. I'd be a little bit cautious there. So I think it's highly likely that Titin does interact with other genes, but we really don't know very much about that at the moment. So at the moment, we really don't know which genes Titin interacts with. We know some of them, but we don't know all of them. And we also don't know the impact Titin mutations have on other mutations. So I'd be really cautious about over-interpreting that data.
Right.
And very.
It's an important point. Yes, absolutely, you're right. We don't know what Titin gene interacts or the Titin protein interacts clearly. but there's 30% patients who are remaining undiagnosed. At least if we know those VUS from other genes, if we can classify them, at least we know, okay, there is a classification of this VUS. Maybe some other patients who will be going through genetic testing has that VUS will be classified automatically. So that's the utility of doing functional assays of these cases with multigenics.
Can you do functional assays for titling? Bruce, how do you do that? Yeah.
So RNA sequencing, we can know. In fact, we have the power of doing, looking at specific mutations in the intronic regions, and based on which intronic region, we can actually have an understanding of whether it's most likely disease-causing or disease-causing based on SAMG guidelines.
So you can assess intronic splices, mutations.
Yes, and also if we get, of course, titin is not expressed in blood, so we have to get the muscle biopsy. So we can do immunoblots, understand the expression pattern, or we can do RNA sequencing.
Immunoblots for titin.
Yes,
doing that?
yes.
Okay. How do you do that?
Sorry?
So titin's an enormous protein. There are very few people around the world that do.
So I'm feeling that we've taken things to a level of complication that might not be useful for most families. So I would like to bring things now that, thank you for this overview. I think it's going to be very useful for many people who are looking for a genetic diagnosis. But I think we need to bring it a bit more to the ground. So the problem with titin, why are you so relatively few people when titin is the largest gene we know? And a big gene is like a big word. When you write a very big word, there's higher chances of getting a misspelling mistake than when you write a very small word. Titin comes from titan. It's the largest gene, it's the largest protein, and that's why one of the reasons why we all carry titin variants. We all carry misspellings in the titin genes, and that's what makes it difficult, because how can we tell which of these misspellings causes disease and which of them is just anecdotal and irrelevant? And that's the challenge we are confronted with. So what Samya was saying is that for some of these misspellings, there are specific assays that allow to assess the impact of this misspelling on the expression of the protein, but I think we need to be very clear about this. This is a minority of the misspellings.
I mean, there are a lot of grays area here, so we have to, but at least we have to give the effort to do functional assays to classify all these unknown variants.
So this can be done for a number of these variants, which is already very good news.
Yes.
Yes. But this doesn't change the fact that for many of the variants, we are still confronted with these difficulties, and this might be part of the reason why for so many of you, getting a diagnosis has been difficult, but it's not the full reason. The other reason is also that the clinical presentation can be very variable, and that the first families were identified in 2007, which feels like a long time for me, but it's not a long time for science, so we're still learning about it. We're still learning every day about how the clinical presentation, how do patients with titin mutations look like, and there's a whole range of presentations, and many people here have done a terrific job of understanding how they present, because there are two ways of getting to the diagnosis. One way is one has a muscle disease, gets genetic panels, and the report says there's these titin variants. And what do you do of that? Is this misspelling bad, or is it just anecdotal? And the other way of getting to a titin diagnosis is that you have a muscle condition and you go to a muscle clinic, and then you assess the phenotype, which is the combination of clinical signs that you see and the biopsy findings, and then we think or not the titin is one possibility. So I think understanding, taking all these elements into account is very important for the diagnosis, because we're still learning, and titin is going to keep us busy for a long, long time because it's very complex. So we need to have all the elements in the best, the deepest analysis possible by the most specialized people possible. And here I think it's important to press the point that
it's legitimate that you ask for a referral to a highly specialized center. If you are in Australia, you want to go to see Emily because she knows how to recognize the presentations of Titin. It's not something that will be easily recognized in the local neuromuscular clinic. So it is legitimate to say, look, I understand that what I have is rare. I understand that it is complicated. I understand that you might not know. Who can you refer me to that might know? And I think it's okay to press point. Emily, do you want to? Oh, sorry.
Sorry, if I may add something. I think this is where us clinicians come into play, in addition to a genetic report and a genetic diagnosis, because, and I don't want to be negative, but you also don't want to be labeled with the wrong diagnosis. I think before zooming ahead and burning steps, common things are still common, and it is extremely poor form to pursue functional analysis on Titin and then end up way down the line recognizing that it's a more common disorder that is more easily or readily diagnosed and which may have clinical trials going on or different monitoring assays or things like that. So I think it is important to put all of the pieces of the puzzle together and to keep an open mind. And by keeping an open mind that's two-sided, the physicians need to keep, and the genetic counselors and the researchers need to keep an open mind, but the patient family, I can't even imagine how hard that could be, but they also need to keep an open mind and not to get too attached to something because we need an answer.
I want to add something to that. Can you hear me?
Yeah.
Yeah.
She's got it.
Is that better?
She's
hello? That's better. Okay, thank you. So I just wanted to say that so we're kind of at the early stages really of understanding the series of titinopathies, and in particular congenital titinopathy that I'm interested in. We've been able to get a really good picture due to the collaboration of a whole lot of clinicians from around the world and also a whole lot of families that have been happy to donate their data to our studies. So thank you for that. We now, I think, have a reasonable picture of what the condition looks like, although there's a lot of work to be done in that area to really tease out the details. But getting that clinical match between what we're finding genetically and what we're finding clinically is actually really, really important. And that helps us to make the right diagnosis, to be absolutely as sure as we can that it's actually a congenital titinopathy or another titinopathy diagnosis and not something else. Because it's not the worst scenario, but a difficult scenario is the situation where you see someone in clinic who's been told that they have a titinopathy and in actual fact the data is uncertain or the data actually doesn't match. And I think all of us have had the experience of having to undo a diagnosis, and that's hard for a family and it's also hard for us. So what we do is tread carefully. We just want to be as sure as we can. Sometimes we can't be completely sure. That can be really hard for families, and we understand that. But at the moment we throw as much as we can in terms of our technology and our clinical understanding to be as sure as we can be. So.
But I just want to say there's good news, because now we are at a very different place from where we were 10 years ago. And now we can recognize, we did it in our lab. Emily did a fantastic job of putting together an international series. So we know what congenital titinopathies look like. We still haven't got a full view of the spectrum, because Titin is so big that depending on where your mutation is located, it might look a bit different. But we do know that it's a myopathy that starts generally early in life, that tends to produce contractures, that is, that limbs become stiff and cannot extend fully. So that's pretty constant. It can have or not cardiac involvement. So that's kind of good news and bad news. Bad news because it's heart disease. Good news because it makes diagnosis easier. And I heard that several of you have cardiac disease in the family, which is not necessarily good news, but maybe this is what made diagnosis possible, because it's uncommon to have a congenital muscle condition. And so when we see this combination of a skeletal myopathy plus heart disease, Titin becomes a very good candidate. And then there's the muscle biopsy, and now we know also that there's a variety or range of lesions that we see when we look at the muscle biopsy under the microscope. But we're starting to get a good picture of what we see and how these lesions can be combined. And then there's this, you have a very nice poster over there about what happens, what we see when we do a muscle MRI of the patients. And again, your MRI might not be that typical, but if you happen to have this typical picture, plus a typical biopsy, plus clinical signs that look compatible, we start to be more confident when the genetic tests arrive, when something is found, and in everybody something will be found, okay? If I've never dared doing my genetic testing because I'm sure I have many types in variants. We all carry that. So it's important to have what we call this deep phenotyping, so looking in detail to the patient muscles, the patient's MRI, the patient biopsy, and of course the results of genetic testing to make sense of it, make sure it is tighting and we are not missing another diagnosis, but also not be shy to say this could be a titinopathy.
Just a quick note on that. So, as you need to, we need to dephenotype. Clinical data is very important, but ask whenever, if you can spread the word that, ask your physicians to provide the clinical data wherever diagnostic clinic you're giving, because the diagnostic setup, when we diagnose, do the genetic testing clinical, if we can do the clinical data correlation, so that is going to be a more focused diagnosis, and rather than fishing around. That helps the clinicians, researchers, diagnostic people. So that's what I said, like it has to be a coordinated effort. There are lots of dots that we need to fill so that we can have the complete diagnosis.
And the other thing to note is that the Titan community is small, but but growing, and the clinicians work very well together. So we actually often meet. I live on the other side of the world, but I'm often in contact with clinicians that I know are working in this field for opinions or for channeling variants to people that have additional ways of interpreting those variants. So we very much have a shared dynamic process going on to make sure that ultimately we give you, the families, the right information or the best possible information we can at the current time.
And I think it would be good to make this into a dialogue. So if you have additional questions, opinion, if you want to share your experience of your diagnostic process, please feel free to take the opinion.
So I'm also responsible for the timeline. So this has been fantastic. I wanted to just give you a little overview of this Titan session. So we can ask a few questions now, and then this Titan diagnostic focus talk is going to wrap up, and we're going to segue into talking about cardiac issues and Titan, and that's going to take us into, then we can finish with some question and answers then, and then we'll have a lunch break, and then later this afternoon we're going to have more of a focus on research and hear from some of our researchers that are here, and then tomorrow we're going to have another session on general Titan care. So we do have a few minutes if you guys have specific questions related to this panel discussion about Titan diagnosis. Yes.
I just have a kind of a general question. Is it possible to have the exact same gene variation and have it cause disease in one patient but not another patient?
Yes. That's why we need to get the deep phenotype and have an open mind when we are doing the genetic testing and correlate with that clinical data along with the physician in charge. So it is possible, definitely. We have some experience in addition, like we know that there are siblings with the same deletion, and one of them is not showing the phenotype bigger. So was that your question?
Yeah, would you know what would trigger it to cause it in one thing? Like in the same family, for example, we're just figuring this out among 5 siblings, 2 with the diagnosis, 3 to remain tested. And can we all carry the same variation and only have it cause disease in certain members? And what would trigger? But what would you saying yes? But what would trigger that? Is it like a virus, or is there anything that you know of in here?
It's important to be careful to make sure that a second variation has not been missed, because we have seen this in a number of genes, and we've talked for years of variable penetrance. That means one variant is expressed in one patient and not in another in the same family. And then as we've gone on with better techniques, we have found that there was a second change that was added to the first one, causing disease in some patients who carried both and not in those who carried only the first one. So
just on that quickly, that in DNA testing, exactly that we get one variant and the patient remain undiagnosed. That's where these functional assays come into the play. So when we do the RNA sequencing or some other functional assays, we find those second variant. And the other thing is that when from our experience, when the physician orders does a requisition for the genetic testing, so there is a little bit of gap between communication with the diagnostic clinic and the physician. So that has to be bridged, and maybe patient advocacy groups and patients can tell that. So the physician has to tell, okay, what is the clinical symptoms? What are the so then the diagnostic clinic can actually advise on the what type of genetic testing you need, because that depends on that.
To take a step back. Just go back to a general statement. Not all variants are equal, and we, in general, have a certain idea, and I'm just talking about Titan right now. We have a general idea of what variants are more likely to be culprit or are almost always disease-causing. So you start with that versus other variants that are not as suspicious. They are probably just part of who you are. So that's one of the first steps too when you're looking at a report and when we as physicians are interpreting a report. And then there's a second variant, of course.
So I might just say with the congenital titinopathy in general, there are two pathogenic mutations on the two copies of the gene, and that's what causes the disease. And when we know we have siblings who have inherited the same two changes, in general they progress relatively similarly. There are some exceptions. It's not always quite the same, but it's usually somewhat similar. In the cardiac situation, I work quite closely with Professor Diode Effect, and in Sydney he's got a very significant interest in this area. What we know is that 3% of the population have a change in Titin. So that's 3 in 100 people that would probably possibly, it's called a truncating change. So it's basically a change in Titin that makes the protein much shorter, or actually makes no protein at all in many cases. We know that they're most likely to cause cardiac problems in adults, and they're the sort of changes that we are most worried about in terms of our Titin parents, and particularly our Titin siblings as well, who are carriers. But what we do know is that the penetrance, so the likelihood that you'll actually develop the condition, is not complete. So that means that there are other factors, and we're still teasing those out, that make it more likely that you will then develop the cardiac complication. You're born with the change, but there are additional things that happen as a result of the particular mutation that you've got, or lifestyle factors, or infections that are going to trigger that condition to present itself. And we really don't have a good handle on that. We've got lots of things that we'd like to look at, and we're hoping that families will actually help us look at those very things. And we'll probably talk about more in the cardiac session. But that's a good example of the fact that you might have the same change as someone else, but an additional lifestyle factor means that you manifest the actual clinical condition, whereas the person that shares the same change doesn't. So in some ways, not all mutations present in the same way.
Just in really simple terms, can someone explain the difference between a truncating and a splice? Because I can read the reports, but I don't have a great concept of the difference.
If I can. So truncating variants are, I mean, if you have sometimes deletions of DNA elements inside a gene, or inside the coding region of the gene, so those can be deleted. Sometimes the other question was like comparing to splice variants. So sometimes you have single mutations, like single point mutations, which can cause a splicing defect, which actually can result in deletion of the gene. So that's the difference, basically. But
can I add to that just a little bit as well? So the particular paper that we put together focused mainly on patients with those truncating or splice-like changes, and that's because they're really likely to be pathogenic. We're much more likely to call those as pathogenic than in fact we're almost certain in most cases. Truncating changes cause a spelling mistake in the gene that cuts the read of the gene off, and it produces a shortened RNA, and it either produces no protein at all or a much smaller protein. So that's why we call it truncating. It's because it basically makes the protein in the transcript shorter. And there's a particular mechanism within the cell that actually gets rid of a lot of the very short transcripts that are produced. So that's why we often get no protein at all. So that's a truncating change. And a splice site change is a spelling mistake that alters the way the RNA, so there's a gene that has the recipe, an RNA that basically is reeled off the gene to make the protein, and then there's the protein itself. Splicing is the mechanism by which the right pieces of the RNA are put together, and splice altering mutations mess that up in simple terms. A lot of those splice altering mutations are also devastating at a protein level, so that means that the protein is put together in the wrong way or isn't actually produced properly at all. So those are the two different mutations.
To add on this, splicing is a normal thing in human, like in normal humans, any normal conditions. But when splice variants are those which actually cause abnormal type of splicing. So that's just to distinguish.
And I think we have to wrap it up. But in terms of what it means for the patient, which is I think your concern, these are the mutations of both types that lead us, it's been said, to a shorter protein or no protein at all. And this we are quite confident that they can cause disease. The problem comes when we find a misspelling that changes just one letter in a huge word. And that's the ones that are very difficult to assess, because for those we haven't got any functional testing. So I would say in terms of what do they mean for the family, I would say splice mutations or nonsense mutations or null mutations or truncating mutations, those are the ones we are quite confident with as pathogenic.
Thank you.
Okay, thank you all. Thank you all.